So, hello everyone. So today we are going to talk about the mitochondria. Uh, Both mitochondria and chloroplasts, these are the organelles that produce energy, either for the animal cells in case of mitochondrion or for the plant cells in case of chloroplasts. Both of these two structures are double membrane structures. There's one additional thing in there's one additional stuff that is what we call thylakoids. These are disc-like structures, and these are basically they originated from the invagination of the internal membrane. So that's another additional membranous structure in case of chloroplasts. While in case of mitochondria, we have got two membranes, an outer membrane and an inner membranous structure. So this is a double membrane structure, organelle. Um, the mitochondrion contains inside it DNA, it contains ribosomes, it contains matrix, and all of this stuff lies inside the internal membrane. And it also contains an enzyme which is known as ATP synthase, and it has got two portions, an FO portion and F1 portion. So FO portion, this portion basically remains embedded into the membrane, while the F1 portion, that is found inside the matrix, and this matrix basically fills up the space inside this internal membrane. These two membranous structures, double membranous structures, both of them have got an identical history as far as their origin is concerned. So if we talk about the evolutionary origin of mitochondria and chloroplasts, when primitive life started, then we had in the beginning we had just prokaryotes, and then later they developed into eukaryotes. Both of them were coexisting, and an ancestral cell, which was eukaryotic cell. So this is a theory for the evolutionary origin of both mitochondria as well as chloroplasts. Where did they come from? The mitochondria and chloroplasts. So this is an interesting story. A eukaryotic cell, which was an ancestral cell, this existed together with some bacteria which were capable of oxidative phosphorylation, and some other bacteria which were capable of photosynthesis. So what happened that accidentally a eukaryotic cell, let's first talk about the origin of the mitochondria. This eukaryotic cell basically engulfed or endocytosed a bacterium that was capable of oxidative phosphorylation. So when this bacterium was engulfed, it contained this enzyme, what we call ATP synthase. So these are the ATP synthase molecules in its membrane. When this was engulfed, this was like phagocytosis. So this led to the formation of vesicle. So this was a double membrane vesicle. The outer membrane originated from the membrane of the eukaryotic cell itself, while the inner membrane, this is the membrane which originated from the bacterial plasma membrane. So the internal membrane of the mitochondrion is basically the plasma membrane of the engulfed or endocytosed bacterium, while the outer membrane of the mitochondrion, we just studied that this is a double membrane structure. The outer membrane came from the plasma membrane of the eukaryotic cell, that wrapped up this bacterial cell. Now this ATP synthase became part of the mitochondrial membrane. So this is embedded into the membrane. So previously here the FO portion, the portion that remains embedded into the membrane, while the F1 portion, which was previously the F1 portion was towards the cytosol of the bacterium, and in this case this is now towards the matrix of the mitochondrion. So that's what we just talked about here. So this is the internal membrane of the mitochondrion, which was basically, so what lies inside the outer membrane. If you look at this structure, this outer membrane, this is the covering or the membrane which came from the plasma membrane of the eukaryotic cell, and this is what lies inside this thing. This is the bacterium, and this bacterium is capable of photosynthesis because it contains ATP synthase molecules. And this ATP synthase contains these two portions, FO portion and F1 portion. We will talk about this later in details. But this is how this mitochondrion came into existence, that both the eukaryotic cell and this engulfed bacterium, they started living in coexistence. Same is the case with the origin of chloroplasts. In this case, this eukaryotic cell engulfed another prokaryotic cell, which was again a bacterium, but the difference was that this bacterium was capable of photosynthesis instead of oxidative phosphorylation. Means this bacterium was capable of producing energy from by utilizing some chemicals, glucose in this case, in case of ATP, while the other bacterium, this bacterium was capable of producing energy by the utilization of sunlight. So in the presence of sunlight, this bacterium could produce energy for itself. So when this bacterium was engulfed by this eukaryotic cell, this turned into again a double membranous structure, the outer membrane and the inner membrane. These two membranes, this is the outer membrane of the chloroplast and this is the inner membrane of the chloroplast. So this inner membrane is basically the plasma membrane of the bacterium, while the outer membrane here is basically the membrane of the eukaryotic cell. So this developed into a double membranous structure, what we call chloroplasts. But we said that there's also some other structure, another structure, what we call thylakoids. So these are disc-like structures. These are also found inside the chloroplast. So basically, these thylakoids, they originated from another budding of the internal membrane. So basically, when this bacterium was engulfed, this was wrapped into this plasma membrane from the eukaryotic cell. The internal membrane, the membrane of the bacterium, it started budding towards inside, once again producing some other vesicles. And these vesicles are now known as thylakoids, and the membranes of these vesicles are thylakoid membranes. And this is where ATP synthase is located, which is involved in the production of energy. And these are again the F1 and FO complexes. FO is the portion of the protein, or this enzyme is basically a transmembrane protein. So FO portion is the portion that is embedded into the membrane, and F1 is the portion that resides inside the matrix of the thylakoids. So this is how both the mitochondria as well as the chloroplasts came into existence. So the origin is basically that they're bacteria, and perhaps this is also the reason that these mitochondria are capable of independent replication. So if demand increases, if you need more energy, then your tissue can increase the number of mitochondria per cell, and if the demand for energy is decreased, the number of mitochondria can also decrease inside the cells. And definitely those cells where more energy is needed, like your muscle cells, like your cardiomyocytes, the muscle cells of your heart, they need plenty of energy. So for this region, they would contain a higher number of mitochondria so that they can produce more energy. And the energy production in case of plant cell is dependent upon chloroplasts, and these chloroplasts again contain inside them these thylakoid membranes, which originated from the invagination or budding of this internal membrane. And these thylakoid membranes here contain the ATP synthase, while in case of mitochondria, the ATP synthase exists on the internal membrane. So this is how both mitochondria as well as chloroplasts originated. We would proceed further with the mitochondria, and we will talk about the oxidative phosphorylation. So here's an overview of cellular respiration. This is an animal cell because it contains a mitochondrion. This is the cytoplasm, and this is the mitochondrion. So we know that we utilize glucose for the production of energy. So the first step is when we consume glucose in food, this glucose is picked up by our cells and enters into the cytoplasm. When it enters into the cytoplasm, this glucose is broken down via glycolysis into two molecules of pyruvate. So pyruvate from the cytosol then enters into the mitochondrion, where it undergoes oxidation and is converted into acetyl coenzyme A. Acetyl coenzyme A then enters into the citric acid cycle and is converted into, it's once again oxidized into CO2. And from this citric acid cycle, well, acetyl coenzyme A enters into the citric acid cycle. From the citric acid cycle, some two other what we call these molecules, the NADH and FADH2. So these are two electron carriers. They carry electrons from the citric acid cycle and transport them to the electron transport chain, where oxidative phosphorylation takes place. We will talk about this stuff later. So now we have these four sections. The first section is glycolysis, which takes place inside the cytosol, where glucose is converted into pyruvate. Then pyruvate is transported into the mitochondrion, and all the rest of these steps, these three steps, they take place inside the mitochondrion. So the first one inside the mitochondrion is oxidation of the pyruvate, which was imported into the mitochondrion. Pyruvate is converted into acetyl coenzyme A. Acetyl coenzyme A enters into the citric acid cycle, and two electron carriers, NADH and FADH2, they carry electrons from the citric acid cycle and convert and take these electrons to the electron transport chain. And then from the electron transport chain, the next step is the formation of ATP molecules, which are produced by the ATP synthase enzyme. We will study them in details later. What we need to understand here is these two electron carriers. NADH, this is an enzyme. This is an electron carrier. And this electron carrier, NADH basically stands for nicotinamide adenine dinucleotide. So this is basically NAD and FAD. H stands for their reduced form. So NAD, let's talk about so both of them are basically involved in what we call redox reactions. Redox reactions are a combination of two reactions, the reduction reaction and oxidation reaction. So both would go side by side. So you need to reduce something in order to oxidize something, and you need to oxidize something in order to reduce something. So both of them go in parallel. Let's look at this example. Here we have sodium and here we have fluorine. So both sodium and fluorine, they bind together ionically to form sodium fluoride. How do they do this? There's one additional electron in the outermost shell of sodium. So sodium donates it to fluoride, to fluorine, and then loss of this electron from sodium puts it would put a positive charge on sodium, and acceptance of an electron in its outermost shell by fluorine would add a negative charge on fluorine. So sodium loses one electron and fluorine accepts one electron. So positive charge on sodium and negative charge on fluorine. So the positive charge on sodium basically attracts the negative charge on fluorine, and then they bind together ionically in order to produce sodium fluoride. So in this case, basically, sodium is being oxidized, and fluorine is being reduced. So the sodium is losing its electron, and this is being oxidized, while fluorine is accepting an electron, and this is being reduced. So that means loss of electron is known as oxidation, and gain of electrons is known as reduction. So it means whenever something gains an electron, this is reduced. So here we have got NAD and FAD. So basically both nicotinamide adenine dinucleotide, and FAD, which is fluorine adenine, flavin adenine dinucleotide. So both of them are basically dinucleotides. NAD is a dinucleotide because its nucleotides contain the nicotinamide base and adenine base. So it's a dinucleotide formed by the fusion of two nucleotides via phosphate bonding. So nicotinamide and adenine, these two nucleotides are joined together to form a nicotinamide adenine dinucleotide. And in case of flavin adenine dinucleotide, these are flavin and adenine. So dinucleotide means two nucleotides. So basically NAD exists here in oxidized form, means it is NAD plus, positively charged. That's what we discussed. When sodium is oxidized, it is basically losing an electron. When it has lost an electron, this is positively charged, and now it has the ability to accept an electron. So whenever possible, this sodium, when it is oxidized, it is positively charged, it has lost an electron, it has the ability to carry an electron. So this can work in these redox reactions by accepting electrons and by losing electrons. When it accepts an electron, this is reduced. When it loses it, then this is oxidized. So NAD is basically the oxidized form of this dinucleotide. When this is oxidized, means it still has, this is positively charged like sodium, and now it has the ability to accept an electron. So when it accepts an electron from the citric acid cycle, this is reduced to NADH. So NADH means when it is converted from NAD to NADH, it means it is reduced and now it is carrying an electron. While FAD, flavin adenine dinucleotide, this accepts two electrons. When it accepts two electrons, then this is reduced from FAD to FADH2. So basically both of them are carrying electrons from the citric acid cycle and transporting these electrons to the electron transport chain. NAD exists in two forms. One form NAD plus, nicotinamide adenine dinucleotide, which is the oxidized form, and the second one is the reduced form of NAD, which is NADH. While FAD basically can exist into three or four forms, three forms at least. FAD, FADH, and FADH2. FAD is also known as quinone. FADH is known as semiquinone, and FADH2 is known as hydroxyquinone. So when FAD is converted into FADH2 by accepting two electrons, now this can move in both the directions. So let's assume here we have FADH, and here we have FAD. So if it loses one electron, this will be converted into FADH. So from hydroxyquinone to semiquinone. And if it was FADH, it loses another electron, so that is converted into if it loses two electrons, this is converted back into FAD. So FAD is quinone, FADH is semiquinone, and FADH2 is hydroxyquinone. So FAD now is the oxidized form, FADH is the reduced form, which has got only one electron, and FADH2 is hydroxyquinone, and it has got now two electrons. So this is carrying two electrons, and NADH is carrying only one electron. So this is very simple. Glucose first undergoes glycolysis, is converted into pyruvate. Pyruvate enters into the mitochondrion, where it is oxidized to acetyl coenzyme A. Acetyl coenzyme A enters into the citric acid cycle. NADH and FADH2 carry electrons from the citric acid cycle and shuttle them to the electron transport chain. Now, inside the mitochondrion, ATP synthesis is done by oxidative phosphorylation. Oxidative phosphorylation is the combination of two things. One is the electron transport chain. Electron transport chain is very simple. We'll talk about it later. What is electron transport chain? But this is basically here in this membrane. So there are several proteins. embedded into the membrane. And basically electrons are shuttled from one protein to another protein. We will study this later. And that simply you need to understand that in electron transport chain, the electrons are shuttled from one membrane associated protein to another and then to another. So these are proteins embedded into the inner membrane of the mitochondrion, where these electrons are shuttled from one protein to another protein and then to another protein. This is what we call electron transport chain. And basically these were the electrons which were taken from the citric acid cycle, which took place inside the matrix here. So electrons are picked up by NADH and FADH, by NAD and FAD, and handed over to these proteins, and from these proteins they are shuttled from one protein to another protein. This is what we call electron transport chain. And the second thing of oxidative phosphorylation is chemiosmosis. Chemiosmosis is basically the movement of molecules across semi-permeable membrane and along their concentration gradient. Means there's some sort of channel for the flow of these, or some sort of membrane, and these molecules can move across these membranes, but they always move from higher concentration to lower concentration, and that's what we call chemiosmosis. And basically oxidative phosphorylation, phosphorylation means addition of a phosphate molecule, and oxidative means which takes place via oxidation. So this oxidative phosphorylation basically is the combination of two things, electron transport chain and chemiosmosis, which leads to the production of ATP. What we see here, it is written substrate level phosphorylation at two steps, one at glycolysis and the second one at citric acid cycle, and the third one is oxidative phosphorylation. So there's difference between oxidative phosphorylation and substrate level phosphorylation. In case of oxidative phosphorylation, we have studied that ATP molecules are generated via ATP synthase enzyme. And via oxidative phosphorylation. Via electron transport chain and chemiosmosis. So here ATP is synthesized by the binding of inorganic phosphate with ADP molecules. So when energy is utilized, ATP has got three phosphate molecules, adenosine triphosphate. So in order to utilize this energy, you need to break down one bond among the three bonds. So ATP is converted into ADP and releases an inorganic phosphate. So one phosphate molecule is dissociated and ATP is converted into ADP, and this leads to the production of energy, and that's the energy or the calories that we utilize. Now this ATP is converted into ADP. So in order to make these ATP molecules again, we need to put the ADP molecules, adenosine diphosphate, back into the mitochondrion, where we have got this enzyme ATP synthase into the membrane. So ADP is shuttled from adenosine diphosphate, is shuttled from the cytoplasm into the mitochondrion, and this ATP synthase converts this ADP into ATP by putting together an adenosine diphosphate molecule and an inorganic phosphate. While in case of substrate level phosphorylation, the phosphate molecule is not an inorganic phosphate molecule. It's rather taken from some organic source or from another substrate. And so an enzyme would take a phosphate molecule from another substrate, which was an organic molecule, and then would add it on to ADP molecules in order to synthesize ATP molecules. So some ATP molecules, pretty low, perhaps 2 molecules are produced during glycolysis, 2 molecules of ATP, and 2 molecules of ATP are produced during citric acid cycle, but both of these two are being produced by substrate level phosphorylation. Means the phosphate molecule is coming from another compound which is phosphorylated. An enzyme takes it from this compound and puts it onto ADP molecule in order to synthesize an ATP molecule. While in case of oxidative phosphorylation, the phenomenon is a little different. We will study how these ATP molecules are produced by ATP synthased by ATP synthase in our next lecture. So let's talk about these what's happening inside the mitochondria in a little more detail, step by step. So glycolysis, pyruvate oxidation, citric acid cycle, and then oxidative phosphorylation. So we have studied that glycolysis. We are talking about the mitochondria, so we'll study the details of things what are happening inside the mitochondria. So as we have discussed, there are 4 steps: glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. So in glycolysis, the glucose, this takes place inside the cytoplasm, and glucose is broken down into pyruvate. So 2 molecules per glucose. So 1 glucose molecule is broken down into 2 molecules of pyruvate. Pyruvate then enters from the cytoplasm. Pyruvate enters into the mitochondria, where as we have discussed, that pyruvate is oxidized into acetyl coenzyme A. So when one compound is oxidized, another compound needs to be reduced. That's what we studied in this example. When sodium is being oxidized, then fluorine is being reduced, because when it loses electron, another molecule has to accept, another atom has to accept this electron. So one loses an electron, and another one accepts this electron. So one is oxidized and the second one is reduced. So that's why these are known as redox reactions. So here pyruvate is being this being oxidized into acetyl coenzyme A, and NAD is being reduced. It means this is carrying 1 electron. So it has carried away 1 electron. So these are the electrons which would be shuttled towards the electron transport chain. That's the job of NAD and FAD, that they have to pick up electrons from another compound and transfer them onto the electron transport chain. So the first step where this reduction takes place, this is during pyruvate oxidation. So pyruvate is oxidized to acetyl coenzyme A, and NAD is reduced to NADH. Now this acetyl coenzyme enters into the citric acid cycle and goes through the citric acid cycle and is converted into different types of compounds. We have this detail on this slide. You can read it yourself. This is converted. So acetyl coenzyme is converted into citrate, and then cis-aconitate and isocitrate and so on, and succinate, fumarate, maleate, and oxaloacetate. And during this process, at various steps, so the compounds are being oxidized, and NAD and FAD, they are reduced. So they're being reduced means when, for example, here when alpha ketoglutarate is converted into succinyl coenzyme A, NAD is reduced to NADH. So this is being alpha ketoglutarate is being oxidized while NAD is being reduced, means it has carried away some electrons. So this is a simplified diagram for a citric acid cycle. We are not interested in the details of these chemical intermediates in this cycle. You have, I think you're pretty familiar with this cycle. So acetyl coenzyme A enters into the citric acid cycle, and inside the citric acid cycle, 3 NAD molecules, they are reduced to NADH. They're carrying electrons, and 1 FAD is converted into FADH2. So both NADH and FADH are now carrying electrons. So they have carried electrons from glycolysis, from pyruvate oxidation, and they have carried electrons from citric acid cycle. In case of pyruvate oxidation, it's only NAD which got reduced to NADH, while in case of citric acid cycle, both NAD and FAD are playing their part, and they're being reduced to NADH and FADH respectively. So both of them, the NADH and FADH2, they're carrying electrons, and they can pass these electrons from here, from the citric acid cycle, towards the electron transport chain. So here I have added some definitions as well. Oxidation is the loss of electrons or an increase in the oxidation state by a molecule, atom, or ion. And reduction is the gain of electrons or a decrease in oxidation state by a molecule, atom, or ion. And they would always go side by side. So one atom or molecule has to be oxidized and the second one has to be reduced at the same time. And this is the reason we call them redox reactions. So I think it's pretty clear till now that how electrons have been carried from glucose by these two electron carriers, which are NAD and FAD. Now they will shuttle them to the electron transport chain. And we will talk about the electron transport chain and synthesis of ATP molecules in our next lecture. This you can read it yourself, what's happening here. So let's talk about the yield of glucose. We have got two types of metabolisms, an aerobic metabolism and anaerobic metabolism. So both of these two metabolisms take place in eukaryotic cells. So this aerobic metabolism is the same what we just talked about. Inside the cytosol we have got glucose. This glucose undergoes glycolysis and is converted into pyruvate or pyruvic acid, and this is transferred to the mitochondrion, and inside the mitochondrion pyruvate dehydrogenase converts pyruvate into acetyl coenzyme A, and this acetyl coenzyme A then enters the citric acid cycle where oxidative phosphorylation takes place. So both FAD and NAD, they carry electrons and shuttle them to the electron transport chain. Electron transport chain then helps ATP synthesize ATP molecules from ADP. So this is what we just talked about. So this is taking place inside the mitochondrion. So what is the overall reaction of aerobic metabolism? The inputs here are glucose, oxygen, and adenosine diphosphate molecules and inorganic phosphate molecules. So that's the input. So glucose is coming from the food that we consume, oxygen is coming from the air that we breathe in, ADP are coming from the ATP molecules which were consumed, let's say, and these were the ATP molecules which are consumed and converted into ADP molecules, and then inorganic phosphate. So all of them enter into the aerobic metabolism and are converted into, so these are the products of this aerobic metabolism. What is produced when this entire procedure is completed, what we have at the end is we produce CO2. So that's what we breathe out. So we breathed in oxygen and we produced carbon dioxide. So CO2 is produced as a result of oxidative phosphorylation inside the mitochondrion, which is then taken back to the lung, and then we breathe this carbon dioxide out. In addition, what we produce is water molecules, 36 water molecules, and we have produced approximately 30 ATP molecules. So it's quite possible. So some biology books mention that there is a production of like I don't know, 38 or 36 ATP molecules per glucose molecule. So basically what happens, but this could be a theoretical number, but this is never achieved. And the reason why this is never achieved, you can never produce this number 36 or 38 ATP molecules from one molecule of glucose, is because there are leaky membranes. So some of these ATP molecules can go somewhere where they can be leaked out from the membranes. And the second thing is some ATP molecules are also consumed when we need to here. So some of them are also consumed when we need to import pyruvate into the mitochondrion. So we need to import both of these two things in order to produce ATP inside the mitochondrion. We need to import pyruvate into the mitochondrion, and we need to import ADP molecules into the mitochondrion, and both of these two processes consume ATP molecules. So the net yield, what we have actually produced from one glucose molecule, that number is believed to be around 29 to 30 ATP molecules per glucose molecule. So yeah, so 2 molecules in glycolysis, 2 molecules in 2 molecules in glycolysis, 2 molecules in citric acid cycle, and 28 molecules in or let's say 30 molecules in from oxidative phosphorylation. So in total 34 molecules, but about 4 of them are utilized, and we are left with 29 or 30 ATP molecules. So that's the approximate numbers. So it's not like all the time you're going to get this number. So this is not fixed, but that's the approximate number, that you would produce roughly 30 ATP molecules from 1 molecule of glucose, although in reality a higher number would be produced, but some of them are utilized for the import of pyruvate and for the import of ADP into the mitochondrion, and some of them are lost due to the leaky membranes. So this is what happens normally inside the mitochondrion. On the other hand, what we have is anaerobic metabolism. So this also has its own importance. The number of ATP molecules produced in anaerobic metabolism is pretty low. We see we have got the inputs are 1 glucose molecule, 2 ADP molecules, 2 inorganic phosphates, and this leads to the production of 2 ethanol molecules plus 2 carbon dioxide molecules plus 2 ATP molecules plus 2 water molecules. Or instead of ethanol we can produce lactate. So we will see where do we produce ethanol and where do we produce lactate. But in overall, if we are going through through this anaerobic metabolism, then we are going to produce 2 ATP molecules per molecule of glucose. So the ATP yield is pretty low in this case. And in addition, we are also producing ethanol and lactate. So where do we produce ethanol is during fermentation. We know that we utilize yeast in fermentation processes. So what happens inside the yeast, when glucose is present inside the cytosol, this undergoes glycolysis, like inside the mitochondrion. It's the same phenomenon, and is converted into pyruvate. Now here we have got a different enzyme. In case of mitochondrion, pyruvate is converted into acetyl coenzyme A by pyruvate dehydrogenase, while in yeast, pyruvate is converted into acetaldehyde by pyruvate decarboxylase. So this is going to remove CO2 from pyruvate, and pyruvate is converted into acetaldehyde. Now this acetaldehyde is converted into ethanol by alcohol dehydrogenase, and this phenomenon is very common in your sugar industry. We have heard that sugar industry also produces ethanol. So what they do, they feed the yeast with glucose because they have plenty of sugar available. So they feed the yeast with glucose, and this glucose, the yeast ultimately produce ethanol, but they do not provide any oxygen to the yeast. So the yeast is working under anaerobic metabolism, and this yeast converts, it consumes glucose, it feeds on glucose, and it produces ethanol via this mechanism. Glycolysis inside the cytoplasm to produce pyruvic acid. Pyruvic acid is converted into acetaldehyde by pyruvate decarboxylase. Acetaldehyde is converted into ethanol by alcohol dehydrogenase, and this is the ethanol that we get after fermentation in sugar industry. Where do we produce lactate? This is also interesting. You might have observed that, for example, you're not like accustomed to intense exercise. You suddenly start playing some games. You suddenly start strength training. So what happens? Your muscle was not used to such an intense exercise. So what happens? You might have observed that when we start a game like playing badminton, hockey, football, etc., which are energy demanding exercises, what happens during the first few days, we are like we start breathing faster. Why do we start breathing faster? Because we have discussed that where we need glucose, we also need oxygen to produce ATP. when we are doing intense exercise, our body needs more energy, and this energy comes from the ATP molecules. So the body tries to produce more and more of energy. So it starts consuming more glucose, but in order to convert glucose into ATP, it also needs oxygen. So this is the reason we start breathing faster. But what happens that after some time, our body is not capable of producing the energy that we are demanding from it, because our body was not used to it. It didn't know that suddenly the next morning we would start doing intense exercise. So the body produces, our body used to produce a fixed number of ATP molecules and keep them in reserve for our routine activities. But suddenly, one morning we wake up, or one evening we go to the games, or we go to the gym, and we start playing some sports, or we start doing strength training, and this certainly leads to an intense demand of energy. But our body was not ready for it, so it tries to compensate it by breathing faster, so that more oxygen can be taken in and more glucose can be utilized to produce ATP. But what happens that after some time, the body cannot keep up with the demand, which is coming from the intense exercise. So we're not breathing enough oxygen into our bodies, or not enough oxygen is being transported to the mitochondria. So what happens that this because of the lack of oxygen, this process is shifted, or this metabolism is shifted from aerobic towards anaerobic metabolism. So what happens inside your muscle cells when there's deficiency of oxygen, glycolysis does take place inside the cytosol, because this does not need any oxygen. So glucose is converted into pyruvic acid, and here, rather than pyruvate dehydrogenase, it's lactate dehydrogenase, which converts pyruvic acid into lactic acid in the absence of oxygen, because oxygen is not available. So rather than pyruvic acid being converted into acetyl coenzyme A, it is converted into lactic acid, and this lactic acid starts accumulating in our muscle cells, and this is the reason that the next morning we feel intense pain in our muscle, and that's because of the accumulation of lactic acid in our muscle cells. It also, I mean, we also feel pain because of trauma to the muscle cells, because if you're doing strength training, you might injure some muscle fibers in the beginning, and then they're repaired during the period when you're not exercising. So that's the reason you're skipping a day in between. But what happens in the beginning, when your body was not accustomed to it, it was not used to such an intense exercise, this could not meet the energy demands because of the deficiency of oxygen. And this was the reason we were breathing faster, because our body demanded more oxygen so that it could produce ATP molecules. But the oxygen that we were providing to our body by breathing faster is not sufficient enough. That was not sufficient enough to meet the energy demands of the body. So what body does, it shifts from aerobic metabolism to anaerobic metabolism to produce energy. So how does it produce energy? Is by converting glucose into pyruvic acid, and pyruvic acid is converted into lactic acid, and this lactic acid is picked up by blood and transported to liver, which is where it is converted to glucose and glycogen. So this is the reason that our muscle hurts when we are doing intense exercise, because the metabolism has shifted from aerobic towards the anaerobic process. So there are three different processes. One is aerobic metabolism taking place in your mitochondria, producing lots of ATP molecules. The other two are anaerobic metabolism. One is taking place in yeast, which leads to the production of ethanol. This is generally done in the industry. This is common in our sugar industry. They're producing ethanol by this mechanism. And then we have got the muscle cell, which in case of absence of oxygen, it can produce lactic acid instead of producing acetyl coenzyme A from pyruvic acid, and this lactic acid accumulates in your muscle cells and it causes pain. So what we have studied so far, just an overview. Glucose is broken down into pyruvate in glycolysis inside the cytoplasm. Pyruvate is transported into the mitochondrion, where it is oxidized to acetyl coenzyme A. Acetyl coenzyme A enters into the citric acid cycle. We have studied the details that during the citric acid cycle, the electron carriers NAD and FAD, both during pyruvate oxidation as well, and in the citric acid cycle, both NAD and FAD, they carry electrons from different compounds here, and they shuttle these electrons to from citric acid cycle, they shuttle these electrons to the electron transport chain, where oxidative phosphorylation takes place. And oxidative phosphorylation is the combination of two phenomena. One is electron transport chain and the second one is chemiosmosis, and putting both of them together we call it oxidative phosphorylation. So what happens here, after they transport these electrons from the citric acid cycle to the electron transport chain, what happens afterwards we will study in the next lecture. Thank you very much. If you have any questions, you can post your questions, and I would be glad to answer those questions. Thank you very much.